Good evening, ladies and gentlemen. My name is James Sterling. I'm Provost here at Imperial College. It's my very great pleasure to welcome you all to the 2017 Sir Ernst Chain Lecture. This annual lecture is named in honour of Professor Sir Ernst Boris Chain, whose legacy at the college endures to this day, not least through the Sir Ernst Chain building, Wolfson Laboratories, a building where he and his family lived as well as worked, and which was renamed in his honour in 2002. And I'm particularly pleased to be able to welcome members of Sir Ernst's family to the lecture this evening. Chain is remembered at Imperial for his visionary foresight about the importance of biochemistry, for founding the Department of Biochemistry in 1964 and for holding the chair in biochemistry from 1961 until his retirement in 1976. Chain arrived at Imperial having already been jointly awarded the Nobel Prize for Physiology or Medicine in 1945 while at Oxford. The prize was, quote, for the discovery of penicillin and its curative effect in various infectious diseases, and he shared it equally with Sir Alexander Fleming and Sir Howard Florey. It was Chain who devised methods for isolating, purifying, and concentrating penicillin so that it became usable as a drug. The Sir Ernst Chain Lecture is one of the most important and prestigious events in the college's annual calendar, and I'm delighted that we have this evening a speaker whose reputation befits, indeed more than befits, the occasion. It gives me great pleasure to welcome our speaker, Professor Ada Jonat. Ada Jonat is best known for her pioneering work on the structure of the ribosome. She was born in Jerusalem and after a period living in Tel Aviv, moved back to Jerusalem where she graduated from the Hebrew University with a bachelor's degree in chemistry in 1962. And in 1968, she obtained her PhD from the Weizmann Institute of Science for X-ray crystallographic studies on the structure of collagen. Subsequently, she was a group leader with Professor Heinz Günther Wittmann at the Max Planck Institute for Molecular Genetics in Berlin, and it was there that she pioneered methods to crystallize the ribosome. Since that time, Professor Jonat has had an extraordinarily distinguished career encompassing a number of leading European and American universities, including MIT, Carnegie Mellon, and the Max Planck Institute in Germany. She today works at her alma mater, the Weizmann Institute, as director of the Kimmelman Center for Biomolecular Structure. Professor Jonat's outstanding contribution to science has been recognized by the receipt of numerous awards, prizes, and honors, including the Wolf Prize in Chemistry in 2006 and the L'Oréal UNESCO Award for Women in Science in 2008. In 2009, Professor Jonat was awarded the Nobel Prize in Chemistry, along with Venki Ramakrishnan and Thomas Steitz for, quote, studies of the structure and function of the ribosome. So it is indeed a great honor to welcome Professor Jonat to Imperial this evening. The topic of her talk tonight could not be more fitting. In talking about resistance to antibiotics, Professor Jonat addresses one of the key challenges facing humanity, indeed a challenge that is highlighted as a priority in our current college strategy and one that we are attempting to tackle head-on with our antimicrobial research collective, which operates to consolidate the diverse array of world-class research in this field across Imperial College to address the urgent threat of antimicrobial resistance. Professor Jonat embodies this multidisciplinary approach, and in addressing the origins of her interest in chemistry, she has stated that her curiosity was, quote, not in chemistry, but in nature in general, urging her students interested in a career in chemistry to, quote, go after your curiosity, find the questions you want to answer, find an important question to answer, a fundamental one, suggest to yourself and then to others how you're going to do it, and then go for it. Try to do the best you can, not the best compared to others, but to yourself. This desire to pursue fundamental questions and addressing the most pressing problems facing us is central to what we do here at Imperial, and today we can all learn from one of the very best. So, ladies and gentlemen, it gives me very great pleasure to welcome the 2017 Sir Ernst Chain Lecturer, Professor Ada Jonat.
Good evening. It's a great honor for me to give a lecture that is called the Chain Lecture. I've met Sir Chain, shortly when I was a young scientist, and I have a lot of admiration to him and to Imperial in general. So just to remind you, this is Professor Chain, and he is not, as you heard, he is not alone. His contribution was recognized together with the contribution of Alexander Fleming, Florey, and himself when they found penicillin as an antibiotic and also how to manage to use it. So these are the heroes for me in Imperial. Actually, these are the heroes that are not with us anymore, and I have some that are with us. Professor James Berber that invited me, and Michael Sternberg that is hosting me. And I would like to thank both of them very much, as well as another professor from here, Dr. David Blow, that if I can thank somebody for teaching me what I know in crystallography, this is him. Back many years ago when I was a real young scientist, he was also not very old. He invited me here and he showed me, he let me work with his people in the lab in order to get my fingers hand-on real crystallography. So thanks again. Let's go to the lecture. And the lecture will be about antibiotics, but about specific antibiotics that has to do with the production of proteins. And as I heard that not everybody here is a molecular biologist or life scientist, I decided to say a few words as an introduction. I think you all know that we need proteins, and proteins are within us, and they are performing almost all cellular functions, these functions that are needed for smooth life, like breathing in a human or in mammalians, or like responding to pressures. Actually, proteins are needed for all living cells, bacteria, flowers, cockroaches, butterflies, also human. So some are really helping smooth life, and some are coming up when they are needed for shocks or for other pressures. In nature, I think I need more dark. In nature, structure means function, or function means structure. And I'm using for this a very elaborated machinery, paperclip, that can do its duties when it has a specific structure, specific fold. So these two have been unfolded by a nervous person or somebody like him. And they are the same. They from the point of view of size and size and composition, they are exactly the same. The people, these nervous people, did not put them in acid, did not cut them, did not boil them, just changed the fold a little bit. Enough that they're exactly the same like this cannot do the job. And that's correct also for everything in nature, especially for proteins. Proteins are long chains of amino acids made of beads that are called amino acids. There are 20 types of them, and the fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of the amino acids building it. So proteins are usually over 100, 150 amino acids, can reach 500 or 6. And they are built of 20 types, and the sequence is determining the structure. And let me show you a structure. This is a structure of a typical protein called enzyme that chops its substrate. So here it is. It's made of atoms. The names are shown here. And the fold is such that there is here an active site. I think that you can see it in here. And I rotated it and enlarged it and put the substrate in the active site. And you can see that the substrate fits exactly. And if it was larger, it won't go in. This is no problem. But if it was smaller, it would shake inside and won't bind correctly, and it will not be cut at the same that it has to be. So this whole structure is dictated by the sequence, and the sequence of the amino acids is dictated by the sequence of the gene, of the letters in the gene that is coding for it. So each protein has a gene, or each gene has a protein. Genes reside in the DNA. It's not the whole DNA, but a lot of DNA are genes. And the genes are coding for proteins. Genes are made of 4 nucleotides, 4 letters of the DNA, or the biological instruction language. So you can see it here. This is a common way to show DNA, made from 2 strands that are opposite to each other and held by these ones. These ones are the language, are the nucleotide or the letters of the language. DNA has 4 letters. This is the whole language, all biological language. And they are arranged within these 2 strands, within these 2 double helical structure, by lines, like pairs, like ladders in anything you have to climb on or to connect together. There are 4 of them. I drew them with 4 different colors, 1, 2, 3, and 4, and they're making base pairs. For those that want to remember or remember a little bit more, they are here. Just some more details. This is where the genes are. Here, they are not exposed, and they are not available for anything, any chemistry. So first of all, these are being transcribed to a similar molecule called RNA that is, again, made of 4 letters, 3 of them exactly the same, 1 of them a little different, but can exist as a single helix, a single strand. And this here is available for reading and translation to protein on the machinery called ribosome. Ribosomes, again, they exist in every living cell in huge amounts. In mammalian, there are cells like the liver that can contain 5 to 6 million ribosomes at the same time, working all the time. Even in bacteria, 80 to 100,000 ribosomes function. They are universal, and they function, their main function, decoding and the production of the protein. It means the bonds between the amino acids, which are called peptide bonds, they are making in almost the same way. very similar way, maybe even exactly the same way. This was not shown yet, but almost shown. And they exist in everywhere. They work very, very fast. They can make up to 40 bonds in a second, in 1 second, depending on the complexity of the cell. So between 5 to 40 bonds, and they hardly make mistakes. The mistakes that we can catch, we can observe, are about 1 to 500,000 or 1 to a million. They make more mistakes, but they get rid of them before we as human beings can even notice them. And I just want to go back to their performance that I said a minute ago, up to 40 peptide bonds in a second. I want to share with you an anecdote from my own life. I was a very good student, the fastest in the class. I had to make 1 bond according to procedure in books. Took me 6 hours, single bond. Okay, a huge machinery. So now that we know how they work, we like to compare them to a factory, cellular factory, that gets the instructions here linearly. What we know is DNA. The factory is made of 2 floors. So the top floor is where the decoding happens, the reading of the instructions. The lower floor is where the bond is being made. So the amino acids are brought to the factory by tracks, which are also RNA molecules called transfer RNA. And when there is a triplet here that codes for that amino acid, it will be connected inside the factory. I don't know to draw inside, so please forgive me for drawing it outside. But it will be connected to the growing protein. The track will come out empty. Meanwhile, the messenger, the code, will progress by a triplet. And the whole process takes some energy that I drew as chimneys. That's all. Very simple. That's the way the ribosome works, and it took us 20 years minus 2 weeks to determine it. So in order to, in order to explain a bit better or to make it more clear, we made a little video, right when we determined the structure. At that time, there were 2 more or 3 more structures that were determined elsewhere. And what I did, I collected and connected all of them together, so there is a dynamic in this movie that shows the different functional states. The movie is available in YouTube for the last 12 years, sorry. And nobody so far complained, so I hope it's correct. And what you see here are bacterial ribosomes. So the top floor is the small ribosomal subunit. Ribosomes in the cells exist as 2 subunits, the large and the small. The small is the top floor of the factory. They come together, they associate only when they have to start producing proteins. So here is DNA reaching to the top floor, to the small subunit, and goes into its path. And this is monitored by factors, like initiation factors, but this one is free. Once the DNA is in place, the first tRNA can be bound. It is brought by another factor, initiation factor number 2. And the 2 factors make sure that everything is really in place and correct. Now the factors are happy, they left, and the large subunit can come and associate with the small subunit based on the complementarity of their surfaces. And in order to make it completely accurate, it also makes changes, conformational changes that makes bridges. So there are 13 bridges. Now the active ribosome has been created, and we look at it from the solvent side. small subunit below, top subunit up. The tRNAs are being brought on by elongation factors. These are the tracks. And the ribosome helps, in, out, in, out, in, out, 40 times a second. We took away the large subunit, so you can see the decoding in the small one, and the production of the peptide bond in the large one, and we left only the tunnels through which the protein goes out. And now in the whole ribosome, you can see the tunnel inside, but you can see something that marks where it is. The whole process continues. The protein grows and goes out through the tunnel. And when there is a stop codon, it means a triplet that says protein is finished. Factors like recycling factor and release factors come, and they are shown here as this yellow thing, and replaces the tRNAs. The two subunits can separate. The protein can go out. And the rest of the system is ready for the next job. So in the movie, you saw everything rather fuzzy, but actually we know the position of each and every atom. What I showed you now again is the bacterial ribosomes. Now many, many more structures are known, and the atoms can be shown there. But in short, the small ribosomal subunit in bacteria migrates as 30S, so this is its nickname. It's made of a lot of ribosomal RNA in gray and many ribosomal proteins, each in a different color. To remind you, this is where the decoding happens, and whoever wants some chemistry, it's written here. The large subunit that is making the peptide bond and its elongation, it means it works as a polymerase, migrates as 50S, and also made of ribosomal RNA and many proteins. So in short, the situation is that the top floor, the small subunit, is here shown on top of this lower floor, and the decoding site, the decoding center, is connected to the peptide bond formation site, which is called PTC, by this, what we call tracks, the tRNA. So the tRNA decoding happens here, and it is involved in peptide bond formation here. Just to show you what is tRNA, almost everywhere in all animals and all living cells, tRNA is a double helical L-shaped molecule that has an anti-codon loop. It means free nucleotides that will make a base pairs with the codon in one side, and a 3' end, the other side, that carries the cognate amino acid, which is fully conserved. So this tRNA can be changed between animals and other living cells with no problem, and also the decoding and the formation of the peptide bond can be made by ribosomes from any animal or any cell in any combination that you can think about. Because of the fundamental role played by the ribosomes, many antibiotics target them. So if one wants to get rid of a pathogenic bacteria, best is to stop or to hamper protein biosynthesis. And this is what the antibiotics are doing, almost half of them. Not penicillin, not the one that Chain and Fleming were working on, but another almost half of the antibiotics that are targeting the ribosome or hampering it or hampering some other steps in protein biosynthesis. The natural antibiotics are the weapons that bacteria from one type uses for interfering with cell life of different species of microorganisms. It's a natural product made by bacteria. This shows that the wars did not start by human beings. It started earlier by bacteria. Question is, how do the tiny antibiotics paralyze the giant ribosome? So bacterial ribosomes have a molecular weight which is almost the same for all of them, about 2.5 million Daltons. And antibiotics are about 500 to 600, sometimes they reach 1,000 Daltons. So these little things have to stop the big ribosome, and they do it very cleverly by binding to functional site. So what you see here is the skeleton of the large subunit on the left and the small subunit on the right. And on it, there are balloons, or these circles. Each of them is representing a family of antibiotics. So in the beginning, when antibiotics came into use, about middle of last century, factories liked to improve them, make them better, faster, less toxic. There was a lot of excitement in pharma companies. This changed recently, but first of all, let's talk about these families. So all of these families are bound to functional sites, and just to remind you, this is where decoding happens in the small subunit, bonding in the large subunit, tunneling through the tunnel, although it's almost the end of the production, it's good to stop it even there. And hinging, these are motions that the ribosome does in order to function. And again, in order to make life a bit better, a bit more. I have to apologize. The movies are nicer than what you see here. And the colors are nicer, and I encourage you to look at it at home. But anyway, how is a small antibiotic work? How does it reach its mission? Here is a very small one, adeine, that doesn't let messenger bind to its path. That's all. So it's enough to be small in order to disturb. Second one that I want to show is also working on the small subunit, tetracycline. Oh God, oh God. Tetracycline disturbs the second position of the tRNA. So, okay, let's look at adeine again. Doesn't let messenger bind. And tetracycline, the next one, occupies the position of the second tRNA. One tRNA is not sufficient to make a bond because it's just one amino acid. So here it comes to the position of the second one and occupies it. The next antibiotic works on the large subunit, and it's the first one from ribosomes that went into use, more or less right after penicillin. And it's called erythromycin, and it blocks the tunnel through which protein will go out. So here you see only very, very short protein can be made. And the last one that you will see also works in the large subunit, and it's the cleverest chemically. It just goes into the bond, doesn't let the bond being made. So if you remember, the bond should be made here, and that's it. Just sitting in it. So very clever, isn't it? Now let's talk about what does it mean? What do the antibiotics mean? So ribosomal antibiotics, they all bind to ribosomal functional sites. I showed 4 of them. And there are about 30 types. So 4 you've seen. And we also said in the beginning of the lecture that ribosomal functional sites are highly conserved, almost the same. And I'm sure that you all know that mandatory for clinical use is the distinction between the patient and the pathogen, the bacteria that disturbs it. How does this happen? So even in very highly conserved regions, sometimes there are subtle differences, and I will show you one as an example. So we talk about erythromycin that blocks the tunnel. So I hope you can see. You look into the whole large subunit. This is the tunnel, and the erythromycin is in red, whereas the whole subunit is in green. So it blocks not the whole tunnel, but most of it. Still, it blocks very efficiently. What you can see here is where it sits. So if we cut the whole ribosome, whole large subunit, perpendicular to its, parallel to its long axis, to the long axis of the tunnel, if there is no erythromycin, protein would come out here. It would be made here in the PTC, peptidyl transferase center, and you can see one of the tRNAs here. And if there is erythromycin that stops it here, all this part will not be there. So this is what really happens. And let's look how it happens and what are the rules or the way that things happen here. So we cut the tunnel perpendicular to its long axis. Here is long axis, and we cut it here. And we found a pocket to which all antibiotics that came from erythromycin that were made as a family of erythromycin, which is called macrolides. How do they bind to this pocket? The pocket is made of RNA, just RNA, and each color that you see inside represents one of the antibiotics from this family. And all the results that you can see are crystallographic results. It means experimental, no calculation here. And the pocket is blocked excellently, just very good, just good, but it's blocked. And proteins will not be able to go through the tunnel. All the macrolide family antibiotics, and if you want, this is the main structure of macrolides, all of them interact with one of the tunnel wall components, which is adenine in position 2058. It's A in position 2058. In all eubacteria, in all pathogens are eubacteria. So the interaction between A2058 and erythromycin is shown here, and the affinity in this part is very, very high, and the positioning is very accurate, as if a scientist designed it, just bacterial. Now, ladies and gentlemen, pay attention to this point here, and you will see the difference between bacteria and us. Bacteria in any organism higher than eubacteria. A to G. A to G. That's all. When there is a G here, the distance between it and erythromycin is too short, so there is no binding. That's all. So, second question that I want to talk is resistance, which is the biggest problem now. And resistance to antibiotics is one of the most severe problems in modern medicine. How does it happen? Bacteria develop specific molecular pathways that cause resistance. As an example, using the same mechanism that gave selectivity from A to G can also be obtained by bacteria that can be mutating itself from A to G. That's it. So now they are resistant. This is not the only resistant mechanism, but the one that can explain nicely what happens. So the increasing appearance of multi-drug resistant strain, together with the minimal, actually negligible number of new antibiotic drugs that are presently undergoing development, is becoming a real big problem, a real big worldwide problem. Thus, it seems that we will soon revert back to the pre-antibiotic era. during which disease is caused by parasites or by simple or severe infections were almost untreatable. Maybe tuberculosis is until now a very awful disease, but wounds or surgery or pneumonia, now they are treatable, but the resistance can revert back to the period that they were not treatable. Or, like the World Health Organization says, they call it post-antibiotic era. Doesn't matter pre or post, the idea is no antibiotic. Even the World Bank estimated that up to 3.8% of the global economy will be lost by 2050 of resistance, because of resistance to antibiotics. Something has to be done. But very few new antibiotics are being developed, are in development. Most large drug companies have stopped making new antibiotics. They are still producing the old ones, but they don't look for new ones. They don't invest in new ones because there is a huge mismatch between the social value of new antibiotics and the high expense of creating them. Companies are made to make money, to make profit. Can we combat fully resistance to antibiotic? So in my opinion, it's very unlikely. Because in street words, bacteria want to live, and because bacteria are cleverer than us. Cleverer than us for survival. But if looking at survival, want to live? Please remember, bacteria were on Earth before us. They are here with us. I don't want to talk about after us. Sorry, this. But anyway, as I said before, most pharma companies stopped searching for new antibiotics owing to this expectation for resistance. I repeated it twice because I think it's really important, but I want to go on and talk about our modest contribution. So we thought that companies don't do much, but we will do something in my little lab. So just before this, I think that you all know what is the microbiome. These are the good bacteria that are residing in us, within us. Most of them are eubacteria. They are residing in semi-exposed mammalian organs like guts, ears, lungs, and skin. They are mainly harmless, but changing in their composition can cause diseases, and they themselves can become pathogens. So it's a very important community of bacteria that we should be careful about treating them. So until recently, almost all available crystal structures were of pathogen models. What you see in the literature, almost all, not all, but almost all, until about 3 or 4 years ago, we could not grow, we are not allowed to grow pathogens, so we looked for bacteria that bind antibiotics like pathogens. And we call them pathogen models, and they were found suitable to illuminate common modes of antibiotic action, like what I showed you until now. What they could not do is explain the species specificity of pathogens, which exist. So consequently, we are now determining, actually we determined, the structure of the large ribosomal subunit from a pathogenic bacteria. And we determined it in very high accuracy with several antibiotics. So I'm not going to show you the whole structure. It's not so different from the normal, from the non-pathogenic ones. And I will only focus on lessons that we studied and what is different. So first of all, we learned that it's possible to increase antibiotic effectivity. More potent antibiotics still can be made. And I want to talk about chloromethylines. These are a family of antibiotics that bind to the active site. They are made of a ring. One of them is a natural, and this is the natural one. And then to the ring there is a extension, a C14 extension at C14, and it can be short, long. It can contain other atoms, not only carbon. It can contain sulfur. It can contain rings, double bonds, and so on. Several companies worked on them until recently, but in 2006, different from others, Madriva from Vienna came into it. And I want to show you what happens with this. But for that reason, I would like to show how the active site looks. So what you see here is the large ribosomal subunit, as you saw earlier. And the active site is here. And when we look at it, this is one of the tRNAs, and this is the second. There are two sites, A and P, amino acylated and peptidyl transfer. And here there would be a peptide bond. And the gray around here is the ribosome. So essentially, what you saw here, you can see here in different, I hope that you can see here in different way. The peptide bond formation will happen here. So this is what happens in the active site. Just enlargement that we'll show a bit more. The ribosome is in gray. tRNA in the A site, amino acylated tRNA is in blue. And the P site, peptidyl transferase site RNA, tRNA is in green. And here is peptide bond. This is where it happens. Chloromethylines bind to the active site when it's empty. So just to remind you, this is what we talk about, and this is the way it binds. So if we look at it from top, this is the active site. All the rings, it doesn't matter what are the extensions, all the rings of all this bind the same way. You can see also here together with the tRNA, all the rings in the same way. But the side chains, there is enough room there for them to do what they want, and they bind differently. You can see also here. Each of them binds differently. What is common to all of them can be seen here. All the rings are at the same position, the same way. But the side chains, the only thing that is common to them is that they are making contacts by hydrogen bonds with the active site only from one side of it. All of them, just from one side. What did Madriva do? They added one hydrogen bond from the other side, this one, and extended the potency by 16-fold, just by that. And now they have one that is even better than this and also passed the phase 3. So it's possible. Second lesson is that in principle, it's possible to increase antibiotic selectivity. Here it's in principle, we didn't do it yet, and no company did it yet, but we think that it's possible to make more discriminative antibiotics, and I want to show. So let's look at erythromycin in red, and the pocket in a cyan, blue, you can see it. What you see here, the one side of the pocket, which is the same in 4 different ribosomes, 2 ribosomes of pathogens and 2 ribosomes of non-pathogens, exactly the same, so I drew it only in one color. But next to it, in this side, there is a protein, L32, and this is an interesting protein. We put one on top of the other, we overlaid here L22 in 4 different ribosomes. 2 pathogenic, the purple and the blue, and 2 non-pathogenic, the yellow and the gray. I hope you can see. So until here, they're all more or less the same. But this part here, the C-terminal, exists only in the non-pathogenic. It doesn't exist in the pathogenic. Just is not there in the sequence. 6 amino acids are missing. So essentially, in the pathogenic ribosome, this region is empty. Just there. So if one think about how to do it, erythromycin can be extended and use this free space, but only in the pathogens. It's not free in the other one. So this will be very discriminative and perhaps stronger. I skip lesson number 3. It's less interesting now. I want to talk about lesson number 4, which is, for me, beyond my expectation. Made me really, I'm excited. So the possibility to find novel antibiotics binding sites. So I think that you don't see anything. You're not supposed to. You're just supposed to see many, many lines. And all of them are the sequence of protein L3 in many bacteria. And you can see that the size, the length is almost the same, a little change here. But more important, these 2 changes here. Inside, there is 2 insertions here and here. And when we looked, I enlarged it. We talk about these 2. When we looked at what are these bacteria, they are pathogens. So if we draw it, and I show it on another protein, which is behaving the same, protein L17. So when we overlay all the structures together, the RNA in blue is the same, but the protein L17, the two non-pathogenic are the same, almost the same. The pathogenic are also almost the same, except for this extension here. This is the extension that I showed you in the similar to one that you could see here. This can be a new position, a potential new species-specific target. It's just in one pathogen. This is nice, but it's not as exciting as the next one. Here we overlaid the RNA, the ribosomal RNA. For those that want to know, it's helix 63. You can see that the structures are essentially the same here, and here, and here, and here, except for here. Here there are 9 additional nucleotides. 9 additional nucleotides within 4,500 nucleotides. Nobody paid attention to it. It's less than standard deviation. Even if it happens in 20 places, and even if there are some places that there are no additional but missing. These are really small numbers within this huge list of nucleotides. But when one looks at the structure, the situation is completely different, because here there is a new structural motif that is not in the model. It's specific for the pathogen. So it can be another potential binding site, actually a good potential binding site. We found several like this. And we draw on this big picture, I drew only the additions, not anything else. And you can see the skeleton in gray and the additions in blue. I hope that you can see. Most of them are on the surface. So we made a little movie. You can see it a little better, I hope. And only the additions, and some of them are on the part of the surface that faces the solvent, and some of them are those that face the small subunit. So here is the solvent parts, these ones, and here are the small subunit parts. So we found 25 different positions like this, and we blocked them. And 16 of them, the blockage of 16 of them inhibit protein biosynthesis of these ones. So these sites are not involved in the primary ribosomal activity. They are not peptide bond formation and not decoding. So the pathogens did not understand that they are important. So far, no pathogen contains genes for their modification. They just didn't learn it yet, the bacteria. This means, I repeat, that so far no bacteria assigned vital roles for these sites. And therefore, for now, I don't know how long is for now, I hope more than a decade or two, these sites can be exploited for the design of advanced degradable antibiotics, hence environmental friendly. So I didn't talk about environment and degradable until now, so I'll come back to it. But we can make for this whatever we want to block them. Actually, so far, the inhibition of protein biosynthesis by the newly identified potential sites was achieved by materials, by molecules that are made of nucleic acid or oligopeptides, completely in our hands. They can be optimized in terms of chemical properties, toxicity, antibiotic action, and degradability. So I repeat again degradability, and I want to talk about it for a second, which is lesson number 5, about the environment and ecology. So most of the ribosomal antibiotics are extensions of small organic molecules like this. They are either inside them, the cause of them, or next to them. And these cannot be digested by eukaryotes, not at all, and also not by the bacteria in the microbiome. So they come out, and they cannot be degradable in the environment. So actually what I said until now, these non-digestible, rather toxic compounds are also non-biodegradable, and thus contaminate the environment. They come, when we take antibiotics, we or animals or whatever, even in small amount, they come out of us. They go out to the sewage. They are so small that most of the purification facilities cannot catch them. So they penetrate into the irrigation system. And in this way, they come back to us via the grass that the cows are eating, via the milk of them, or so on. So this is one of the ways that resistance to antibiotic grows even without using antibiotics, just because they're there. So this, if I can remind you, this region here can be used for antibiotics that are degradable. We can do what we think we can from chemical point of view. We don't have to rely on compounds that bacteria already did. So what I said now, the inhibition of protein biosynthesis by the newly identified potential site was achieved so far by molecules composed of nucleic acid and oligopeptides that can be optimized in terms of chemical properties and so on. That's exactly what I said earlier. So in summary, the new insights obtained from these high-resolution structures of the ribosome from a genuine and pathogen provide unique chemicals for several things, for suggesting novel sites, for potential future antibiotics I just showed, for suggesting how to improve chemical performance I showed, for better distinction between pathogens and useful bacteria, I think that I also showed, so for producing pathogen-specific antibiotics, which means design of antibiotic drugs specific for each and every pathogen, in contrast to the current preference to use broad-range antibiotics. So we are aware of the fact that pharma companies will not be happy about it. They are even not happy about making broad-range and pathogen-specific antibiotic is even less profitable. We expect that even pharma companies will consider human aspects, not only profit. Actually, there is here also a profit point of view, because if antibiotics are out of use and infectious diseases will come back, then the companies will not have enough customers for their expensive everyday usage drugs, or for drugs that are made for bad diseases of elderly people. So I don't want to go into more detail. I think you got the message. And when I tell it to companies, they don't like to hear it, but they have a second thought. Okay, so efficient use of pathogen-specific antibiotics means fast identification of the type of the relevant pathogen. When a patient comes to hospital, which pathogen is it? But the doctors told me that steps towards this aim are being made. It also means that it means the use of pathogen-specific antibiotics. It means that we have to know, to identify the unique structural features of each and every pathogen, which means many structures. How will we get many structures? By X-ray crystallography? So the first structure, as I told you, about 20 years, and then later 2 to 4 years, we have no time for many structures of such length. So luckily, recently, three-dimensional cryo-EM became useful, and we think that this will be the way to show, and in order to prove to ourselves that we can do things, we first of all looked at the ribosome of the parasite Leishmania, eukaryote, and we saw that within a few months we had a real nice map of 2.8 angstrom. I just want to show you where Leishmania is active, which is a large part of the world, not including Britain, but including many other countries. And I just want to show you that we got the structure of the ribosome, the large ribosomal subunit, all the proteins, and all the rRNA, ribosomal RNA, without having sequence. We had to sequence them too within a few months. Here are all the proteins that are in the large subunit. I really don't want to go one by one, but I want to show you the quality. So you can see here side chains and so on. And I want to show you the whole RNA, which could show differences between purines and pyrimidines and so on. We could also see modifications like methylation, dihydrouridine. 99 modifications we could identify, although there was no literature about it. So it's possible. For those of you that are interested in the resolution, it's shown here. I don't want to talk about it, but I want to tell you about another lesson. Until now, everything was fine concerning EM, but when one really wants high detail, not always EM is okay. Just real quick, that we have structures that were determined by us at about 3.5 angstrom, and by cryo-EM, a little bit less resolution, but most of, at least formally the same, more or less the same, a little bit worse. However, we did find, like the EM, that the two antibiotics that we didn't know about before, avilomycin and varomycin, are binding in the same way, the same place. But we, crystallographically, could find every specific hydrogen bond that these antibiotics are making. I don't want to describe them, but we could understand the resistance to it and the selectivity. Whereas the EM people wrote that they need better structures for it. So not always. I want to do a lot of cryo-EM, but not always. In short, our structure-based species-specific design aim only at minimizing. Don't think that we have an answer to all the problems. Minimizing widespread resistance, microbiome destruction, and environmental hazards. So essentially, I only have to thank people here and acknowledge my group and so on. But Mike asked me to talk about history. You still want it?
Yes.
It will take a few minutes. So before I go, another, sorry that I didn't say it earlier, another take-home lesson from our studies is that there is a bright future for antibiotics and for environmental and hazard and microbiome problems. How did we start? So what was known when we started, that proteins are cell workers and they are made according to the genetic code by the ribosomes. In 1980, in January 1980, or actually in December 79, I said that I want to understand the structural basis for ribosome function. I was not the only one, but I said it. And since other people said it before me and failed, most of the scientific community, in a very polite way, said, you must be kidding. It means they were laughing at me. But they said it this way. And I said that I want to understand the interior. It means really how it works. They said that these are black boxes. I don't think I have to say here that crystallography is the only way to understand, or was at that time the only way to understand or to visualize structure at atomic resolution. But maybe I will just show quickly. Oh, good. Doesn't work. So crystallography needs crystals. And you can see here salt crystals. Salts are only 2 atoms, and they really love to order themselves periodically in 3 dimensions. So this is a crystal thing that you can see. This, when you buy salt, you buy it already spontaneously crystallized. Also, proteins can be crystallized, and there are some people here that worked on lysozyme, the TIR protein, crystallized it in Oxford, and a kid, say 6th grader, could repeat it. But those, now that we know how they build, even at that time that they didn't, those can stand so nicely in line and also in 3rd dimension. There was a big question mark because of the failures of the others, which was actually no. And the explanation for it, the way that those that try to crystallize and could not, so they consider the ribosomes not crystallizable, explained it by their high degree of internal mobility, which is all correct, considerable flexibility, correct, functional heterogeneity, correct, chemical complexity, and large size, all correct. I thought, though, that they overlooked one of the reasons, or overlooked or didn't give enough attention to it, and this is a marked tendency to deteriorate. When people produce ribosomes, less than half of the preparation is active, even in the best laboratories in the world. 40–50% activity is called very good. Happened to me too. But I thought, what happens to the other half? The half that is not. It's just floating about there, half deteriorated or not. I thought that this is the reason why they can't be crystallized. The preparation is 2 or 3 days. The crystallization may take weeks. If in 2 or 3 days already half of them are gone. Okay, so this is what I thought. And I was lucky I had a possibility to read some things that are not directly science, or not at the molecular level at least, and this is about bears at their winter sleep. So it was found that during the bear's winter sleep, the ribosomes are orderly packed in monolayers inside the part of their membrane that looks into the cell, inside the membranes. So I read that, and the delegation that reported it didn't think it's a big discovery, but anyway, they wrote it. For me, first thing, it showed that ribosomes can be orderly packed. Scientists couldn't make it, but every bear, every cell, every winter, it must be possible. And I thought, why is it so? Why? Why does the nature provide this type of mechanism? And in my opinion, when the bears get up at the end of the winter, they want to do all types of things that they need proteins. And for having these proteins, they need ribosomes. So I thought that the periodic dense packing is meant to, or is the mechanism for maintaining a large quantity of active ribosomes during the 3-4 months that they are not needed. And this brought me to the suggestion that stress induces periodic packing of ribosomal particles. Stress doesn't have to be only cold temperature. It can be any stress, in my opinion. So the solution actually was, and you can read only these 2 lines in green, using ribosomes from bacteria that live under stress, like in the Dead Sea or the hot springs, or waste of nuclear facilities. So for those of you that didn't see the Dead Sea, this is the way it looks from top, from closer. All this is salt that came out when the sea evaporated. You see just evaporation of one summer. You can see that although it's called Dead Sea and no tree, no animal here, there are 2 types of bacteria in the sea. And here, this is one. You see the pinkies here? These are colonies of Heloarcula marismorti. Marismorti is Dead Sea in Latin. And Heloarcula, Helo shows that there is a lot of salt here, 3 molar KCl. Just incredible. So you can see also here the colony. And I knew a little bit about this bacteria. I crystallized some proteins from it before, so we tried these ribosomes. We also tried ribosomes from hot springs, as I said before, and from Deinococcus radiodurans, which is a bacteria that can live under super extreme conditions. Very hot, very cold, hunger, dust, and irradiation. Can be even irradiated. So this, and it really behaves like pathogens, except that it's not. So the first microcrystals were obtained half a year after we started. For those of you that are interested, experiment 29, that contained 25,000 different conditions, but only 29 experiments. Anyway, we got beautiful crystals after a few years. And after 6 years, we decided that it's time to go measure them. And we went to a synchrotron. For those of you that don't know what is a synchrotron, it's an accelerator. I show here the one in Grenoble, although it's not the only one. And it sits here. Inside, it's made of a booster where the particles are being accelerated and then run around in a circle. And on the tangentials to the circles can be stations that are designed for different experiments. Here is one of them, the ribosome, measured by Frank. So we got there in 1986, 6 years after we started, and we got fantastic diffraction. Beautiful. You had to hear the young people, yes, yes. And I was there already 6 years, so I did even twice as yes. But not for long, for just one exposure. So we repeated this experiment 100 times in different synchrotrons all over the world, in America, in Europe, and so on. And always we could not, sometimes we could get 2 exposures, and then the whole thing deteriorated. So if you allow me, a little bit emotions. Before that, we started here, and we saw only this mountain, and we called it Everest, and we started climbing on it. Climbed and climbed and climbed. 6 years until we got to the summit. And what did we discover then? That there is a bigger Everest, that now we have to get there and who knows how. So I don't want to go into all the details, but we thought that ribosomal crystals are readily damaged by X-rays because there are, in every damage, there are two steps. But primary is it's the break of the bonds, and the secondary, the propagation of it, that when a bond is being broken, the two parts of the bond, the two atoms are unhappy and they run around. And the ribosome crystals are relatively empty, a lot of solvent, so they can run around, look for electrons or get rid of the electrons and break more and more bonds. So we thought that we cannot stop the first step. The beam always will cut, but the propagation we can minimize by reducing the temperature. And the breakthrough actually came by flash cooling at cryotemperature. This became a routine right away, but I want to show you the experiment. It was in Stanford. The experiment is less important. What's important is my face, you see? You see that I didn't even believe in this experiment, but it worked. We collaborated with Hakon Hope from Davis, and look how beautiful. Great diffraction and the structures you have seen. You can see me 20 years later in Grenoble with the machine that keeps a cold stream, a cryo stream. I look a bit more optimistic. So except for me, during the time, okay, let's say it differently. Until we did the experiment, which is right here, there were hardly any structures. Since then, all that. 99% of them were determined by cryobiocrystallography. This is actually the number of PDB, protein data bank deposition, number of structures in the world, and this is the year. I must say the two more advances were made when we introduced the cryocrystallography, MAD for phasing, and better detectors. So the three things together brought us now to 2017, with about 85,000 structures. Most of them are of materials that otherwise couldn't be measured, and most of them of materials that are important as the medicine or for new materials. So now I can give credit to those people that first of all allowed me to work. Michael Sella, the president, who was an immunologist, and everybody said he let this crazy dreamer, fantasionaire, it means me, because he's a biologist, but he was replaced by Chaim Harari, a physicist who didn't change it. And this is because they were encouraged by the Scientific Advisory Committee that was chaired by Sir John Kendrew. I guess that you know his name here. Chris Anfinsen and Alex Rich. Alex Rich himself tried to crystallize ribosomes for 10 years together with Jim Watson, that you should know his name too. They had the institute for it at Caltech, and they failed. But anyway, although they failed, Alex was very supportive. It all started with a very intensive collaboration with Professor Wittmann from the Max Planck for molecular genetics in Berlin, who, you know, a word about Wittmann. He said that he had a group of 245 people. It means several groups, but this was his department. And he's very, very open. He let every group work on anything that they think that is scientifically sound and important, as long as it's ribosomes. So there we started. He was a fantastic collaborator in terms of knowledge, ribosomes, people, and in establishing a research unit for us at the synchrotron in DAISY, which functioned until 2004. we'll show in a minute. But he died 10 years before the structures came, and then he was replaced by Franceschi and then by Fucini. So I had two groups working in parallel, and I want to thank the group members for their devotion and determination in good and actually more in bad times. I want to show you the group in Hamburg that, as I told you, was closed down in 2004. The technical assistant said that nobody can work on ribosomes, only angels. And the rest of the group that came to the Dead Sea to look for better bacteria. You see how they have a great time. The group in Rehovot, in Weizmann, is still functioning. I show here what's going on the last 3 years. It means some of them have already left and some are there. But what's important for me to show is that I'm there only, I don't know how to describe myself, but the real leader is Dr. Anat Bashan. She's running the show all the time, the students and so on. I will not tell you what everybody did, but I will focus on Tamar. Tamar came for 10 weeks, 16 years ago, and she's still around. The day that I took the picture, she had birthday, so she made a cake. And this is her cake. One thing is shows for the young girls here that it's good, that it's possible to be a good housewife and a very good scientist. And the second is that in my group, ribosomes are considered sweet. I also want to thank my family, my sister, mother, daughter, and granddaughter. The daughter is now an MD, and the granddaughter, when she was about less than 9 years old, I got this prize in Paris, UNESCO prize, and she wrote a speech for me. My name is Noah, Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. Girls, it's possible to be a scientist and a housewife or grandmother. She gave me a prize. No, not this one. This is the Nobel medal. But her prize for me is as important, maybe even more. And this is the Grandma of the Year is Ada Yonat. And the nice thing about the Grandma of the Year is when I ask her which year, every prize has a year. And she said, which year? You have to reprove yourself every year. And if you fail, I take it off the wall. I want to show you it's still on the wall. Ribosomes became popular. So here is a carnival. He is me, and she is wife. You see, she's a ribosome. They're dancing. Protein comes out, and young children became ribosomes too. And this is what happened to me. by an artist that is not a scientist, and he never met me before. And after 5 years, I met him, and I said, how did you make it? He said, just from the internet, small subunit, large subunit, you see? And he thinks that new antibiotics should be here. So thank you.
So thank you very much for an amazing talk. We've got time for 1 or 2 questions. And I'm reminded what the previous lecturer said, that the best questions are short questions. So if anyone would like to ask other something. At the back.
Sorry, I'm probably the first away. I'll try and shout.
No, we've got mics, otherwise people won't hear.
Even better. So you mentioned about how you want antibiotics to be biodegradable because they can enter the food stream. Is there any real hard evidence linking the antibiotics being in the food stream and the emergence of resistance?
Do you understand the question?
repeat the question.
Sure, yeah. So you said you would like antibiotics to be biodegradable because they can enter the food stream and potentially emergence of resistance can occur. Is there actually any hard evidence linking the emergence of resistance directly from the environment rather than the misuse of them?
No, the main, okay, I got you now. The main resistance is not from the sewage, it's from the use and because it is a natural process that bacteria make in order to exist. But the sewage or the environmental problems are not making it better. So whatever we can do to minimize, we do. About direct evidence, I think yes. I think that as far as I remember, there was a whole study showing how chicken, in chicken's blood or in chicken's, how you call this thing?
Guts.
Yeah, guts, stomach. There were pieces, the cores of antibiotics that were never given to them.
Okay. Thank you.
So there's a question just over there as well. The mic doesn't have to travel far.
Hello, I don't know if this is related at all to your research, but in certain industries, for example, the oil industry, there's a lot of bacteria, different types of bacteria, and what they're trying to do is fight bacteria with different types of bacteria. But the problem is if your, the type of bacteria that you're using to, let's say bacteria B to fight bacteria A, is in short supply due to logistics, then bacteria A flourishes much more than it would have done. And is this a dangerous way of looking at things and combating one bacteria with another bacteria?
So let's look at it from the positive way, positive point of view. There are studies that talk about bacteria against bacteria, the balance, the amount, and so on. And I'm not a microbiologist, especially not a counting microbiologist. And they have several outputs. And one of them is that bacteria can stop other bacteria from growth. And the idea is to raise those bacteria that are less dangerous for us. But more than this, I can't tell.
Thank you. It's actually, they use bacteria that's less dangerous for metals because of corrosion, rather than humans.
Thank you for a terrific talk. I'm intrigued by your antibiotics by design. And I wondered what you thought about, in particular, the design of antibiotics for pathogens which are non-bacterial, so like eukaryotic pathogens. Given that most eukaryotes have a little microbe within their cells, the mitochondria, which has a bacterial-like origin, do you think there is going to be more success in designing antibiotics that target the mitochondrial ribosome or the pathogen cytosolic ribosome, which is, I guess, more like ours in terms of specificity?
So far I know about antibiotics against some, not pathogens per se, but poisoning or, how you call it, like malaria, leishmania. They're all working on the part of ribosome that is the same in the cytosol and in the mitochondria. Most of them are aminoglycosides. And some of them were designed for completely different reasons, like genetic diseases. And they are good for these parasites. If the parasites suffer more in the with antibiotics in cytosol or in mitochondria, I really don't know, but I know about human. When aminoglycoside is prescribed for a cytosolic reason and it penetrates the mitochondria, it can lead to problems in hearing, in in seeing death or blindness. So one has to be careful about that.
Okay, so I'm conscious of the time, so it's an incredible pleasure and privilege to be able to give a vote of thanks to Ada. Politics has been described as the art of the possible. And I think possibly we could say for Ada, science is the art of the nearly impossible. I first met Ada when she came, I was running the group in the Weizmann and visited Professor Phillips' laboratory at Lysozyme, and that had 1,000 atoms, and crystallographers at the time were solving 1,000, 2,000. The tour de force, I think, was actually in Oxford from Louise Johnson, who did something like 7,000 atoms. The next time I met Ada was when I visited friends in Berlin in about 81, and they told me, you remember Ada, she's now trying to solve the structure of the ribosome. And what she said is, with the general consensus, this is completely impossible. You saw the graph of the number of protein structures going up. There was that one or two structures, and they're all small. And now Ada was trying to solve something, it was 2 orders of magnitude larger. Anyway, the impossible became possible, and as we saw, Ada with others won the Nobel Prize. Looking over the information, Ada was the first lady in the Middle East to win the Nobel Prize, and possibly shocking...
Not such a big deal.
Okay, well, it is. And shocking, even more, is that she was the first female to win the Nobel Prize for chemistry for the last 45 years, which is just an incredible period of time where there hasn't been a female Nobel Prize winner. So, it was an amazing talk. I greatly enjoyed the movies showing the molecular basis of antibiotic resistance. And I think the one slide still that really captivates, I think, the excitement of structural biology is seeing that 2 atoms changes, and it's the difference between life and death. The antibiotic working, the antibiotic not working. And that's 2 atoms in 250,000 atoms. And I think that really encapsulates the excitement, I think, that we both share for structural biology, and you've conveyed that to the audience. And really, I'd like to take this opportunity to thank you very much for coming all the way from Israel to give the Ernst Chain lecture, which clearly antibiotics, from Sir Ernst Chain, antibiotics from Professor Ada Yonat. Thank you very much.